Good afternoon. My name is Justine McKinnon, and I will be reviewing my CILE topic on the spread of fluoroquinolone-resistant Campylobacter and alternative antimicrobial managements at the herd level, a narrative review. For several years, both veterinary and human medicine has relied on antibiotics for various treatments, all while starting with the simplest antibiotics, including beta-lactamase antimicrobials such as penicillin. However, antimicrobial resistance has become a major public health concern globally as antibiotics are continuously used for growth promotion purposes. Furthermore, the continuous use and misuse of antibiotics amongst food-producing animals is common worldwide for nonspecific uses, including both prophylactic and metaphylactic treatments. Thus, research has determined that various antibiotic classes, including fluoroquinolones, have developed various mutations, including mutation and acquisition of antibiotic resistance genes affecting both DNA gyrase and DNA topoisomerase 4, which are two major drug targeting enzymes that have further influenced native efflux pumps in the bacterial membrane, leading to selective resistance of Campylobacter species, including Campylobacter coli and Campylobacter jejuni, which is the leading cause of zoonotic foodborne related illnesses in food animal populations, including poultry. Thus, several studies have indicated drug-resistant strains of animal origin are common and easily spread through various routes, including the food supply chain, direct animal contact, or through a contaminated environment, all of which have indicated that antimicrobial resistance has been linked to intestinal microbiota in livestock commonly carried by commensal bacteria through studies showing the selected antimicrobial resistance genes can easily be transmitted to various pathogens through horizontal gene transfer. With genetic variations occurring constantly, horizontal gene transfer has since led to the development of new resistant gene variants and genetic material to arise in bacterial populations through various mechanisms, including transformation, transduction, or conjugation, allowing for commensal microbes to acquire new genetic material outside their colonial lineage, allowing for further expansion among other bacteria. Thus, with Campylobacter species studies focusing on ciprofloxacin and nalidixic acid resistant strains, this has shown that even with limiting or completely banning the use of various fluoroquinolones in food animal populations, such as within the European Union and the United States, selective resistance has resulted in single point mutations known as C257T in the gyrase A gene within the quinolone resistant determining region, ultimately leading to a synergistic effect amongst fluoroquinolone efflux pumps, allowing for continuous circulation of Campylobacter resistant strains to remain in the environment for years. Thus, studies determined that contaminated fecal material containing antibiotic resistance genes is the biggest reservoir for selective pressure development, as it is quite easy to transmit between species via direct contact through the water supply and within the environment. Globally, this is a widespread problem amongst both developed and developing countries, as individuals tend to be in direct contact with these particles, or that the manure is often used as fertilizer for crops and other necessities, regardless if pre-treatment procedures are performed in an attempt to decrease bacterial contamination. Furthermore, ciprofloxacin and nalidixic acid-resistant Campylobacter strains amongst food animal populations tend to contribute to problems involving foreign travel to developing countries, as many of these countries have no laws governing the use of antibiotics in food animals, leading to continuous spread of antimicrobial resistance genes through horizontal gene transfer. With the continuous threat of antimicrobial resistance, a multi-facilitated approach to various alternatives involving multiple management practices, including sanitation, waste management, high nutrition practices, disease surveillance, and biosecurity, have been recommended and are implemented to help reduce the effects of antimicrobial resistance worldwide. The World Health Organization and other affiliations, including the OIE and the FAO, have since created the Global Action Plan on Antimicrobial Resistance with 5 major objectives, including improved awareness, involvement of surveillance systems, reduction of infectious disease, optimization of rational antibiotic usage, and the mobilization of research and resources. With attempts at maintaining antimicrobial resistance at the herd level, the current literature has shown that even with various surveillance systems, including the National Antimicrobial Resistance Monitoring System in developing countries, these systems are complex and will take time to improve. Furthermore, efforts must be made in providing surveillance abilities in developing countries, as having the ability to obtain proper data collection is a key factor in providing more research-based evidence to reduce and control the spread of antimicrobial resistance. Furthermore, improved biosecurity measures must be improved by promoting good agricultural, hygiene, and veterinary practices amongst those who are in direct contact with livestock, along with promoting intensive farming systems to slowly convert to extensive and/or organic-based extensive systems, which can help reduce and maintain lower levels of antimicrobial resistance amongst the livestock and the environment. Lastly, veterinarians must also play a role in the control of antibiotic usage by limiting antibiotic usage by prohibiting unrestricted access to antibiotics amongst farmers, and that these antibiotics are only available through a veterinary prescription, as this can reduce the prevalence of antibiotic resistance gene spread, leading to selection pressure and potential cross-resistance of bacteria. In conclusion to this literature review, due to the numerous gaps in scientific evidence thus far, more research-based evidence is needed to determine if these alternatives can help globally in reducing antimicrobial resistance. However, it is essential that we continue to improve and provide national surveillance systems in monitoring antimicrobial resistance, as these systems can aid in improving and promoting research-based evidence in an attempt to control and limit the spread of antimicrobial resistance from food animals to consumers. To end this presentation, I wanted to emphasize the importance of the subject matter and other public health concerns around the world. Besides pursuing my master's in public health, I am also pursuing my degree in veterinary medicine. Over the years, I have worked in many aspects of the veterinary field in both domesticated, small, and large animal settings as a licensed veterinary technician. Thus, it has given me several glimpses of the similarities between veterinary and human medicine, and how our environment provides a link between both humans and animals. Thus, being given the opportunity to pursue my master's in public health, it has allowed me to gain a hands-on approach to the ever-growing multidisciplinary field of public health, which can help me as an upcoming medical professional to collaborate between various professionals both in the veterinary and medical fields. Thus, my passion is not only focused on the numerous fields of veterinary medicine, but also the opportunity to focus on the public health tract with special interest in zoonotic disease and antibiotic resistance aspects, which would also allow me to build on interactions with various officials and the public on the importance of public health. Lastly, this program will assist me in achieving future goals of becoming a practical veterinarian who will utilize a One Health approach in both veterinary and public health settings. Thank you for your time.